So thank you all for coming today and having us here visiting Stanford and UCSF. I'm David Strauss, and I'm here with 5 of my colleagues, Wendy, Ziwa, Ksenia, Robert, and Daryl. And within the next 45 minutes or so, you're going to quickly hear from all of us. So I'm going to go ahead and get started. We're going to talk about moving new science into the regulatory review process, a focus on cardiac safety of new drugs. So I am right now the acting director of the Division of Applied Regulatory Science, and 4 of us are from this division that are here today. And our vision is to move new science into the FDA review process and close the gap between scientific innovation and medical product review. We perform research, mission-critical applied research to develop and evaluate tools, standards, and approaches to assess safety, efficacy, quality, and performance of drugs. And we also perform expert regulatory review consultations for mechanistic safety and other critical scientific questions for immediate regulatory needs. And this can combine critical review of existing knowledge, computational analyses with informatics tools and disease pharmacology models, both in vitro and vivo laboratory studies, and translational analyses combining preclinical, clinical, and post-market data. I'm going to quickly highlight a few areas of research that we perform in our division and highlights broader types of research at FDA. The first topic being modernizing toxicology and safety pharmacology with humanized assays and genomics. A second, bioanalytical pharmacokinetics and drug-drug interactions research. And the third, informatics tools for regulatory review and mechanistic safety. And for the rest of the talk, about 80% of it is going to focus on this first topic, the Cipa Initiative, Comprehensive In Vitro Proarrhythmia Assay, a cardiac safety initiative. And in the last talk, Daryl Abernathy is going to discuss systems toxicology and pharmacology applied to cardiac toxicity in addition. So the problem we're talking about today is drug-induced torsade de pointe, which is a arrhythmia that can cause sudden cardiac death. And it had long been recognized that many cardiac drugs can cause this arrhythmia, but in the 1990s it was realized that we had a, quote, epidemic of drug-induced torsade with non-cardiac drugs, and many were withdrawn from the market as shown in this slide. You can see they're across all different types of indications, not just cardiac drugs. And it was realized that drugs that cause torsade block the hERG potassium channel, a specific ion channel, and prolong the QT interval on the ECG. So in response, there's an international body called the International Conference on Harmonization, which seeks to harmonize global regulatory guidelines and requirements for drug development and registration. And it consists of the major regulatory authorities around the world. And they developed 2 guidance documents. The first one's a non-clinical guidance, and it focused on measuring the specific ion channel block, the hERG channel, and non-clinical animal action potential duration, or QT interval studies. And the second was the clinical guidance, which focused on measuring a drug's effect on the QT interval. And it set a threshold of concern that was very small, approximately 2% of the baseline QT interval. And this led to it becoming the most intensive and expensive of the clinical pharmacology studies in drug development. And because of this low threshold leads to a certain number of false positive findings. But what have the results been? After implementation of these guidelines, no drugs with unrecognized torsade de pointe risk have been approved or removed from the market. So it's been successful in that regard. But it's occurred at a cost. And that's been that there's been a premature discontinuation of drugs from development due to hERG or QT signals that is sometimes due to an inaccurate perception of risk. Many potentially good drugs never get evaluated in humans due to a hERG effect. And many drugs with QT labeling are unlikely to be proarrhythmic. We now have a deep mechanistic understanding of the ionic factors that result in torsade de pointe. And this can support a new approach to assessing proarrhythmic risk beyond hERG block and QT prolongation. And this has led us to the Cipa Initiative, the Comprehensive In Vitro Proarrhythmia Assay. Its goal is to develop an in vitro and an in silico computational paradigm for cardiac safety evaluation of new drugs that provides a more accurate and comprehensive mechanistic-based assessment of proarrhythmic potential. And over the next 4 talks, my 4 colleagues are going to give an overview of these components and the research we are performing at FDA to support this. And before handing this over to Wendy Wu, I'd like to highlight that while we have a large effort related to this at FDA, this is a true global effort. We have multiple nonprofits helping coordinate involvement from the global regulatory agencies and industry and academia.